Good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next and every exam. It's about time for NECO 2025 Biology Practical. Now, for this forthcoming practical, we've been given 15 specimens. And having looked at these 15 specimens, as you can see on the board, we've been able to look at the links, the probabilities, and all of that. We've put the 15 specimens into two groups. The first group talks about those in A to G. There is this link in there. And then the second talks about specimen H to O. We'll be looking at the probable question in the exams. Okay. And at the same time, we'll be looking at probable drawing. So the plan is this: for specimen A to specimen G, we'll look at that in a lecture video. For specimen H to specimen O, we'll look at that in a second lecture video. And in a third lecture video, we'll be looking at the probable drawing. So how we start, how we end, how we label, and all of that. So let's get right into it. The first question says that identify specimen A and B without reason. Of course, specimen A and B, as you can see in the diagram on your screen, are housefly and spider. Moving on to question 2. Question 2A says that name the class of animals to which specimen A and B belongs. Of course, when you talk about classification in biology, you remember we usually start our classification with kingdom, phylum, class, order, family, genus, and species. So we're told to look for the class of specimen A and specimen B. Now, for specimen A, the phylum of specimen A and B is that they belong to the phylum Arthropoda. And the class of specimen A is Insecta, while the class of specimen B, the spider, is Arachnida. Now, if we move further about the classification, if we were told to state the order of specimen, order to which specimen A and B belongs, specimen A, talking about the housefly, is actually a diptera. The reason why we say diptera is that, look at it, di means two. When you talk about housefly, it has two wings. So it is two wing or true flies. They actually do fly. That is how they live their life. So that is why it is classified as diptera. When you talk about Arachnida, why are we what is the order of Arachnida? That is Arachni. And the reason for that is because they breathe via the trachea or the buccal lung. So that ends question 2A, talking about the class of specimen A and specimen B. Moving on to the next question, that's 2B. We should give reasons for the class stated in 2A. So the question is this: why are we putting specimen A into the class Insecta, and why are we putting specimen B into the class Arachnida? Okay. And the reason we are putting the housefly into the class Insecta is because it has three body division, as you can see in the diagram on your screen. The body is divided into head, thorax, and then abdomen, three body parts. It has three pairs of legs, three pairs of walking legs, and that's a common feature or characteristic of the class Insecta. It also has a pair of jointed antennae. Yes, for sensitivity, it has antennae, and it also has a segmented abdomen. When you look at the diagram on your screen, the abdominal region towards the ending part is segmented. The body is divided into various equal segments. Then now, when we talk about the class Arachnida, why are we putting spider into the class Arachnida? The reason is very simple. It has two body divisions, cephalothorax and abdomen. When we say cephalothorax, the word cephal is from the word cephalos, which means head. In this case, thorax means the chest region. So the head and the chest, they are fused together. That's for the spider, you can see in that diagram. And then you now have the big oval abdomen. So it has two body division. That is why we are able to put it into the class Arachnida. Also, it has 4 pairs of legs on the cephalothorax, 4 pairs, not as against 3 pairs which insect has. So which means these reasons we can also use it to differentiate between specimen A and specimen B. Now also why we are putting this into the class Arachnida is because it lacks antennae. Also, it has one pair of pedipalps. You see it in the diagram on your screen, and it has simple eyes, that's ocelli. Whereas when you talk about the insecta, it is majorly compound eyes. So that answers question 2b as to why we are classifying A and B into the class Insecta and Arachnida. Moving on to question 3, it says state economic importance of specimen A and B. Okay, what are the economic importance of specimen A and B? In fact, we could also add something like specimen E to that, talking about the economic importance. You can see those questions on your screen. Let's look at economic importance of specimen A and B, but we can add that of specimen E to it because of its importance in pollinating flowers. As you can see on your screen, economic importance of specimen A, housefly, it transmits disease. When you talk about housefly, it is the vector of viruses, bacteria, and protozoans, which causes polio, cholera, typhoid fever, yaws, and dysentery. 2, it causes food contamination. 3, it causes food deterioration. Talking about the economic importance of housefly, you see, when you talk about housefly, it is able to regurgitate vomit fluid from the crop to moisten solid food. This brings viruses and bacteria picked up from feces and other places into food. That is why we talk of food contamination there. Housefly also defecates constantly on food. That is why it is able to contaminate food and cause food deterioration. We've been able to state 3 economic importance there. Also, when you talk about specimen B, you can see on your screen what are the economic importance. Spider is a form of pest control. When you have spider around, they predate on insects. You can see that on your screen. They are predators of insects, which are crop pests and can help reduce the need for chemical pesticides. You see, insects are pests to the crop. Now, spider feeds on these insects, so that way the spider becomes a pest control. Also, in some cultures, spiders are consumed as food. So spider is a form of food in some cultures. Also, number 3, as you can see on your screen, it is an important part of the food chains. It's serving as prey for some animals. For example, birds feed on spiders. Lizard also feeds on spiders and other animals. So that is talking about completing the food chain, where the spider now, when it feeds on insect, is a consumer, but the spider now is serving as a prey for other animals like birds, lizards, and the likes. Now, if we were to have economic importance of, let's say, something like butterfly, what would be the economic importance? It's the adult butterfly is a very useful insect because it helps to pollinate flowers during its visits to flowers in search of nectar. When you talk about nectar, nectar is the sugary fluid that we have in flower. You can see on your screen, the adult butterfly is very useful insect. It pollinates flowers during its visit to flowers in search of nectar. Pollination brings about fertilization of flowers, leading to production of seeds and fruits. Also, the larvae of a butterfly, referred to as caterpillars, feed greatly on the green leaves and young stem, destroying them during the process of feeding. That way, the photosynthetic activities of plants is greatly reduced. Also, in trying to control caterpillars, that's the larva of butterfly, a lot of money is spent on buying chemicals to kill them, pesticides, insecticides, and all that. So those are the economic importance of butterfly, and that answers that. Can we also have like economic importance of grasshopper? Economic importance of grasshopper is that they damage crops by eating leaves and roots. Grasshopper here is beneficial because they help to aerate the soil. Once they burrow into the soil, they free the soil particles, allowing air to come in, so they aerate the soil, and also they add to soil fertility when they of course pass feces onto the soil, so they add to soil fertility. So that talks about the economic importance of specimen A and B precisely. We are just adding that of specimen E and specimen G. That's talking about grasshopper by design. And that ends question number 3. Worthy of note is the concept of metamorphosis. You know, in the development of an animal, of organisms, there is all called metamorphosis. It is the series of gradual changes that occur during development of an animal from zygote, that's from the moment when the egg is fertilized till the animal grows to the adult stage. And we have two types of metamorphosis: complete and incomplete metamorphosis. As you can see on your screen, for complete metamorphosis, you have from the egg to the larva to the pupa to the adult stage, specimen A and specimen E. That's talking about housefly and butterfly exhibit complete metamorphosis. They go through 4 stages: egg, larva, pupa, adult. Now for the incomplete metamorphosis, you're looking at the animal moving from the egg stage to the nymph and then to the adult. Grasshopper exhibit incomplete metamorphosis, and that's just about that. Moving on to question 4, it says state 3 observable features of specimen E and G that helps them adapt to their environment. So it means that what are the characteristics or features that E and G possess that enables them to adapt to life in their habitat or environment? Talking about the butterfly, what are the features it possesses that makes it adapted to its way of life? Okay, one is that it has large and broad wings for flight. It has compound eyes for good vision. It has long or thin proboscis, that's mouth part. You can see it in the diagram on your screen. That proboscis is used for sucking nectar. Nectar is a sugary fluid secreted inside a flower, you know. It also has a long clubbed antenna for sensitivity. Those are the features that adapt butterfly to its way of life. So we're talking about grasshopper. How is it well suited for its mode of life, that's to its habitat? The first is possession of antennae for detecting food. The antenna here is for sensitivity. The antenna for grasshopper is for detecting food. Compound eyes for wide view. Wings also for flight. The mandibulate mouth part, that's it has mandible mouth part for chewing and cutting, where that of butterfly is for sucking mouth part. This is a chewing mouth part. That is why your grasshopper is able to chew on leaves of plants. Also, it has chitinous body cover or exoskeleton that protects the soft tissues on the inside. So that is how specimen E and G are well suited or adapted to their habitat and way of life. And that ends question 4. Moving on to question 5. Question 5 says state 4 observable structural differences between specimen A and B. Observable structural differences between specimen A and B. So we go back to specimen A and B. What are the observable structural differences that we can see in specimen A and B? As you can see on your screen, specimen A, talking about the housefly has wings, the spider has no wings. The housefly has antenna present, spider has no antenna. The housefly has 3 pairs of legs, spider has 4 pairs of legs. Talking about the housefly, it has 3 body divisions: the head, the thorax, and the abdomen. Whereas for the spider, it has 2 body divisions: the cephalothorax and the abdomen. What we mean by cephalothorax here means the head and the thorax, that's the head and the chest region, is fused together as one. And you can see that in the diagram on your screen. The spider just has a smaller top part, that is the cephalothorax, the head and the chest region already fused together, and then the big oval abdomen. Okay, so those are observable structural differences. Also, for the specimen A, the house fly, the mouth part is a mandibulate mouth part. For the spider, it is a non-mandibulate mouth part. You wonder how we are able to see the mouth part of the housefly and spider. In this practical, we were told to make available a hand lens or a magnifying lens. So if you view specimen A and B with a hand lens, you will see that for your housefly, it has a mandibulate mouth part. For your spider, it has a non-mandibulate mouth part. The housefly has compound eyes present. For the spider, compound eyes absent. 7. Abdomen is segmented for the housefly. The abdomen is divided into repetitive segments, but for the spider, the abdomen is not segmented. It is just a very airy, big oval abdomen. So that's just about 7 differences, structural differences. That's differences in the structure of A and B that you can see. So that ends question number 5. Moving on to question 6, you can also see that on your screen. It says we should state structural similarities between A and B. What are the similarities structurally that we can see between housefly and spider? When you look at both possess simple eyes. The housefly, in addition to the compound eye, also has simple eyes. When you talk about the spider, it has simple eyes, about 8 of them, called ocelli. Now, when you look at the legs of both animals, both have hairy walking legs, which ends in claws. That's the legs has hairs on them, which ends in claws. Those are just about the 2 structural similarities that we can see. Moving on to question 7, it says we should state the specific habitats of specimen A and B. Specific habitats of specimen A and B. When you talk about the housefly, as you can see on your screen, the housefly, you can have them in latrines, toilets, refuse dump, exposed food, sewage, and decayed organic matter. For our spider, we have them in forests, deserts, rainforests, rocks, mountains. We could also have specimen E, F, G to that. In what habitat can we have them? For specimen E, talking about the butterfly, we can have them in the garden, the farm, flowering plants, trees. When you talk about F, the crayfish, we have that in fresh water, like rivers, streams, lakes, ponds. The grasshopper, we have them on grassland, forest, farmland, and vegetation. And that just about answers question 7. Question 8 says, make a labeled drawing 12 to 15 centimeter long of specimen B. We should make labeled drawing of specimen B. That's talking about the spider, 12 to 15 centimeter long. Okay, talking about the drawings for this exam, we are going to have that in a separate lecture video. Like, link to that particular lecture video on the drawings for this NECO 2025 biology practical is in the description of this lecture video. So that ends question 8. So moving on to question 9, we were told to identify specimen C and D. Without reason, identify specimen C and D. So specimen C is pride of Barbados flower. You can see that on your screen. And specimen D is bean seed. Now to question 10. For question 10, it's quite a handful. Okay, it says name the floral part of specimen C. Indicate the number of floral parts in each wall of specimen C. Talking about 10 Roman figure 1 and Roman figure 2, you can have the question just like you have on your screen, or you can as well have a table, where you will be told, okay, what you should name the floral part of specimen C. That's pride of Barbados flower. You should tell us the number of the floral parts that you have, the color of those floral parts, and should state maybe, those floral parts are essential or non-essential. You can see in the structure of flower that you you have your pistil, stigma, the stamens, the anther, the pollen grain, the receptacle. You also have the calyx at some points there. But straight to the question, we're told to name the floral part. Of course, we have the sepal or calyx just after the receptacle. It is green. It protects the delicate part of the flower. It's towards the base of the flower. You have the sepal or calyx, and depending on the flower you're given for or at your specific center, the sepal or calyx, you have the petals or the corolla. The petals or the corolla are the bright, colorful, a light flowery wall. So petals or corolla. You have the stamen or the androecium. This is the male part of the flower, from where pollen grains are released, and then it gets to the stigma or the pistil, or what we generally call the gynoecium. That's the female part of the flower, so that the pollen grain gets to the stigma and move out the style to the ovary, where you have the ovule, so that fertilization will take place. So these are the floral parts of specimen C. They have the sepal or calyx, petals or corolla, stamen or androecium, the stigma or the pistil, or you call it the gynoecium. So how many number? What is the number of these that we have on specimen C on that flower? It could be 5, it could be 4. Check well when you get to your center. The sepal or the calyx usually 5 or 4, as the case may be. The petals or the corolla usually 5 or 4, as the case may be. Depends on the flower that you are given. Then when you talk about the stamen or the androecium, look very well at the diagram you have on your screen. The yellow part, that is actually the stamen, or the where you have the stamen or the androecium, where you have the pollen grain. That's the tip. There is the anther. So when you say the stamen is a combination of the anther and the filaments. They make up the stamen. So it is from this stamen, that's from the anther, that pollen grain is released. Okay, we usually have a good number of them. It could be 10, it could be 8, it could be 9, so very numerous. Just count them. But the one at the center is now the stigma or the pistil or the gynoecium that receives the pollen grain release from the anther or the stamen or the androecium, as the case may be. So it's usually one at the center. So what is the color of the floral part? The sepal or the calyx is green. The petals or the corolla, depending on the flower you're giving, it could be red, it could be pink, it could be yellow. The stamen or the androecium could be red or pink or yellow. As you can see in the diagram on your screen, this one here is yellow. Now the stigma or the pistil, the one you have at the center, that's the gynoecium, is usually red. It's usually red at the center. So when you look at the sepal and the calyx, the sepal or the calyx, and then the petals or the corolla, the sepal and the petals, they are the non-essential parts. In fact, we can be told to remove all of the sepal and the calyx, and we should be left with just the stigma and the stamen. It depends, because you see the essential part of the flower is actually the stamen and the stigma, the male and the female parts for sexual reproduction in flowering plants. So essential, essential, non-essential, non-essential, and that just about answers question 10, Roman figure 1 and Roman figure 2. So we can be told to state the functions of the floral part. What are the functions of the floral part? The sepal, the petals, the stamen, the stigma. As you can see on your screen, the sepal protects the developing bud. It protects the delicate flower structure from drying out. When you talk about the petals, it is to attract pollinators. It is to attract pollinators. You know, the petals is very bright colored, very attractive. It is to attract insects. It is to attract butterfly, those that will help to pollinate, to carry the pollen grain from the anther to the stigma. Then when you look at the stamen, what is the function of the stamen or the androecium? It is to produce or release pollen grains. That's the male part of the flower to release to produce and release pollen grains. Then the stigma is to receive and capture the pollen grains during pollination, thereby initiating fertilization. So we can be told to state the functions of the floral part. Moving on to the next question, question 10, Roman figure 3 says, name the sex of specimen C and give reason for your answer. We should name the sex of the Pride of Barbados flower. Of course, because it has the male and the female part, it is an hermaphrodite, or you say bisexual. And the reason is this: it has the male and the female parts. That answers that. Also, question 10, Roman figure 4 says, what is the symmetry of specimen C and give reason for your answer. The symmetry of specimen C is bilateral symmetry. We can cut it into two equal halves along one plane. So the symmetry of specimen C is bilateral symmetry. Is question 10, Roman figure 5. It says, name one pollinating agent each of specimen C and D. That's for specimen C and specimen D. We should name one pollinating agent. Now, for the flower, the pollinating agent is insects such as butterfly, honeybees, and then for the bean seed, the pollinating agent is wind. To Roman figure 6, it says we should state adaptive features of specimen C that attract the agent of pollination. That is, those pollinating agents of specimen C. What are the features that specimen C possess that attract those pollinators, that is attracting those insects? What are the features or characteristics that specimen C has that attract insects or those agents of pollination? 1. Brightly colored flowers or petals. 1. Brightly colored flowers or petals. Then 2. The petals or the flowers are conspicuous or large. Then 3. Nectar. The flower secretes a sugary rich liquid called nectar, which the insects see as food. In fact, even butterfly, when they get there with their proboscis, they suck the sugary liquid called nectar as food. So that is why those insects are always coming to the brightly colored flower, not just for the color alone, but for the fact that they are going to get food. Also, colored pollen grains attract them. That, as you look at from the diagram on your screen, the pollen grain yellow, brightly colored. Also, the surface of the flower is rough, and when you look at the stigma, it is sticky surface for pollination. That is why those agents are coming. Also, the petals smells good. They are scented. The petals scents good. They are scented. You say scented petals. So those are the adaptive features of specimen C that attracts the agent of pollination. And that just about ends question 10, Roman figure 6. To Roman figure 7, it says make a label drawing 8 centimeter to 10 centimeter long of the longitudinal section of specimen C and label fully. You can see that on your screen. We want to talk about the drawing of specimen C. In another way, it goes like using a scalpel or knife or razor blade provided, cut through specimen C longitudinally, dividing it into 2 equal halves. Make a label drawing of 8 centimeter to 10 centimeter long of 1 half. Like I said, the link to the lecture videos on the various drawings we have for this exam is in the description of this video. You do well to check that out. Moving on to Roman figure 8, it says name the placentation. You know, from question 7, we must have used the scalpel or the knife to cut specimens, to cut the flower longitudinally. Therefore, we should be able to see the placentation. If you look at question Roman figure 8, VII, it says name the placentation that we have on specimen C. The placentation is marginal. When you look at the ovals, they are marginally arranged. They just like the margin of your book. When you look at the ovals inside the ovary, they are marginally arranged. So the placentation is a marginal placentation. You can see that on your screen. Placentation of specimen C is marginal placentation. They are marginally arranged. To question Roman figure 9, it says what types of ovary does C have? What type of ovary does C have? It is a superior ovary. Ovary is positioned above other floral parts. You look at the ovary. When the ovary is positioned above every of the other floral parts, is above the receptacle, is above the sepal, is above the petal, you know, then you say it's a superior ovary. So look at the question again, IX. What type of ovary does specimen C have? It is a superior ovary. That is to say, the ovary is positioned above other floral parts. So moving on, question 10, Roman figure 10. Question 10, Roman figure X. Now, it says, what are the observable features of biological interest of specimen C? We can also have that question. It says, what are the observable features of biological interest of specimen C? It means that what are those features that specimen C has that are of biological interest? You can see the answer on your screen. Sepals are brightly colored. It could be yellow, it could be pink, it could be white, it could be red. One of the petal is modified into tube-like structure fold. Honey guide or standard petal. Okay. 3, large or fan-like brightly colored petals. Long brightly colored filament. You see the anther is brightly colored. That's talking about the stamen, the pollen grain, the pollen. They are brightly colored. Long brightly colored filament, you can see that. So those are those features that are of biological interest to specimen C. It's about the color and the structure, you know. That answers that. Then question 10, Roman figure 11, XI, it says mention 2 elements contained in the class of food produced by specimen C. Mention 2 elements contained in the class of food produced by specimen C. In there we have carbon, we have hydrogen, we have oxygen, we have nitrogen, we have sulfur, we have phosphorus, because the class of food produced in there is a protein. Mention 2 elements contained in the class of food produced by specimen C. And the answer is very simple. The class of food is actually protein, and the element there, carbon, hydrogen, oxygen, nitrogen, sulfur, or phosphorus, just speak about any two elements, and that just about ends that. Moving on to question 11, it says suggest the food substance present in specimen D. Now, the food substance present in specimen D is protein, and in Roman figure 2, question 11, Roman figure 2, it says what function does it perform in the body? So what are the functions of protein? Recall that specimen D is being seed, so it says what food substance is present there? It is protein. What are the functions of that protein? We need protein for growth, we need it for repair of worn out or damaged tissues, and we need that also for the production of enzymes. Remember that most reactions in the body are catalyzed by enzymes, so proteins are needed for the production of enzymes. 11 Roman figure 3 says to what class does specimen C and D belong? Specimen C and D, that's Pride of Barbados, flower, and the bean seed. To what class do they belong? They are legumes, or you simply say leguminosa, or you just say Magnoleopsida. By saying Magnoleopsida, it means they are dicots. There's two seed leaf dicotyledons. Okay, that answers question 3. To what class does specimen C and D belong? Legumes or leguminosa or Magnoleopsida, that's their dicot. Now to Roman figure 4, it says mention one agricultural importance of the class mentioned above. So agricultural importance of legumes. One agricultural importance of legumes, not biological importance now. Agricultural importance is the fact that legumes make nitrates available to the plants, that's in the soil, and consequently the plants. You see, legumes have root nodules which harbor nitrogen fixing bacteria like Nitrosomonas and Nitrobacter. Nitrosomonas and Nitrobacter, which converts ammonia into nitrites and nitrates respectively. Okay, possession of root nodules which harbor nitrogen fixing bacteria that makes nitrates available to the plant. Of course, you know, nitrate is nitrate is one of the macronutrients required for plant growth. So that is the agricultural importance of legumes. They make nitrate available in the soil and consequently the plant. And with that, we've come to the end of this lecture video talking about specimens A, B, C, and D. So we will be discussing specimen E, F, and G in the next lecture video, quite a handful. And for myself and the entire team, it's bye for now.